Yeah, good day everyone. You're welcome to today's practical class. Today we shall be looking at the flower. The main thing we're going to look at the flower as the reproductive part of a plant, especially the reproductive parts of an angiosperm plant. We're going to look at the parts of a flower. We see that we're looking at the stalk, which is also called the pedicel. We're going to look at what we mean by the thalamus or the receptacle, the floral parts. We identify all these floral parts and we'll be able to look at the walls, the two walls, the first two walls, the calyx and the corolla. Then after looking at all or identifying the parts, we are going to draw the whole flower. Then what you are going to do, or how you are going to do or go about it, is you go outside, look at a tree, the flower, you pluck the flower. Make sure that the flower has clearly defined parts. That means the parts are fine, the parts are completed. You bring that to the lab, you observe the flower, you identify the parts, and you draw a diagram of 8 to 10 centimeters long, and you label the parts. Then the next activity is how to dissect a flower. You plug another flower, you make sure that this flower is also having its parts clearly defined. You bring that to the laboratory, you observe the flower very well, and you try to identify or point out the parts of the flower. You use your razor blade to cut the flower longitudinally, that is downwards from top down. When you do that, the flower is expected to produce 2 equal parts. You take one part, you draw and label fully. Then another thing you are going to do after that is that you can also take another flower, remove the petals and the sepals, use your blade to cut out the stamen, and you cut the stamen from top downwards. Use your magnifying glass to observe, then you draw and label what you see. Then for the third diagram, you are going to use your razor blade to cut the female reproductive parts. You cut it longitudinally into 2, that is the stigma. Observe what you see with the magnifying glass, then you draw and label what you observe fully.